Howdy, my name is Adamari de la Cerda, and my partner Mario Flores Castro and I will be discussing our second experimental lab report on the types of bacterial culture media. To begin, bacteria are microorganisms that need certain conditions to grow and thrive. These conditions include temperature, moisture, pH, and nutrients, to name a few. Culture medium helps provide some of these conditions for bacterial growth and may be solid or liquid. With that said, the purpose of this experiment is to understand what culture media is and its role in bacterial growth. When conducting this experiment, the bacterial cultures needed are E. faecalis C, A. faecalis, C. perfringens, E. coli, K. pneumonia, S. typhimurium, S. flexneri, S. aureus, S. epidermidis, S. pyogenes, and of course the unknown samples, in this case 101 and 201. Additionally, the agars and mediums needed are 6 blood agars, 5 CNA blood agars, 5 mannitol agars, 5 McConkey agars, 5 Salmonella Shigella agars, 4 motility mediums, 5 bioesculin agars, 5 EMB agars, 4 blood agar plates for the aerotolerance test, and 5 thioglycolate mediums for the aerotolerance test as well. In this experiment, each solid agar or medium broth were aseptically inoculated using the listed corresponding bacterial cultures with a loop or needle. Additionally, each plate was labeled accordingly to their inoculated bacteria and a corresponding medium before being sealed and incubated. For the aerotolerance test, two blood agar plates were divided into three sections and inoculated with the corresponding bacterias seen at the bottom left of the slide. Another two blood agar plates were divided in half and inoculated with the unknown bacterial samples as seen on the right hand side. In the end, one trio and one of the half plates were placed in an anaerobic chamber while the other two remained in a student basket. Lastly, five thioglycolate mediums were inoculated with the listed bacteria and unknown samples.
CNA agar is useful to isolate gram-positive bacteria from a variety of specimen types. In the plate for S. aeres, we can see how there was bacterial growth and the colonies look small and yellow. This is as expected from S. aeres as this type of bacteria is gram-positive. From the unknown 101 we can see how the cultures look big and cloudy. In S. pyogenes there was little growth, as the same with E. coli and the unknown 201. Mantle agar plates are useful so that we're able to differentiate between bacteria that are able to survive in areas of high salt concentration from those who don't. And here we can see how S. aeres and unknown 201 showed growth in the plate, while S. epidermidis, the unknown 101, and S. pyogenes had no growth. It is expected that S. aeres will have growth, as S. aeres is a staphylococcus which is able to survive in high salt concentrations. Salmonella and Shigella plates contain bile salts that inhibit the growth of gram-positive bacteria. Therefore, this type of plate is useful to able to grow gram-negative bacteria. And here we can see how unknown 201, E. coli, S. diffimurium, and S. flexneris has growth. McConkey agar plates are useful so that we're able to isolate gram-negative bacteria from a variety of bacteria. And here we can see how E. coli, S. diffimurium, and unknown 201 show bacterial growth, while S. aeres and unknown 101 did not. S. aeres is gram-positive, and that is why there was no growth, while E. coli and S. diffimurium are gram-negative, therefore that's why there was growth on the plates. Blood agar plates are useful so that we're able to identify bacteria that are able to break down red blood cells in a process known as homeolysis. And here we can see how unknown 101, unknown 201, and K. pneumonia show homeolysis, while S. aeres and E. faecalis there's no homeolysis present. In S. pyogenes, there is no bacterial growth shown. Bile-squalene agar is useful so that we're able to differentiate Enterococcus from Streptococcus. And here we can see how in E. faecalis there is a black complexion on the plate, telling us there's growth, since E. faecalis is an Enterococcus that is able to grow in the bile of the plate. S. pyogenes there is no growth, as S. pyogenes is a Streptococcus. In unknown 101 and unknown 201 there wasn't any growth. EMB agar plates are useful so that we're able to grow gram-negative bacteria. It also helps with the differentiation between lactose fermenting and lactose non-fermenting bacteria. And here we can see how unknown 201, E. coli, K. pneumonia, and Staphylococcus show bacterial growth. E. coli, Staphylococcus, and K. pneumonia are gram-negative, therefore that's why there is growth. Unknown 101 shows no growth.
The motility test medium is considered a semi-solid agar that determines a bacteria's motility. This type of medium contains low agar concentrations that allow motile bacteria to diffuse away from the inoculation line, while non-motile bacteria only grow along the line. From the results of this experiment, bacterial diffusion is seen from K. pneumonia, E. coli, and the unknown sample 101. From this, we can determine that these bacteria are motile. On the other hand, the unknown sample 201 displays a profound bacterial growth along the inoculation line. This reveals a possibility that the bacterial unknown sample 201 may be non-motile. Although a bit of K pneumonia displays a bit of non-motile growth, it is not as profound when compared to the results of unknown sample 201. The thioglycolate medium is used for the cultivation and separation of anaerobes, aerobes, and microaerophils. The results from this experiment can be interpreted by analyzing turbidity that may display varying growth patterns depending on the organism. The results from this experiment display the possibility of facultative anaerobes in unknown sample 101. Additionally, the unknown sample 201, C. perfringens, A. faecalis, and E. coli might indicate the presence of microaerophils that can thrive in lower levels of dioxygen. The aerotolerance procedure tested selected bacteria for any oxygen requirement necessary for growth. Most notably, C. perfringens does not show any growth in a blood agar plate where oxygen is present, as seen on the top two pictures of the slide. On the other hand, A. faecalis, E. coli, and the unknown samples 101, 201 can thrive with oxygen in the blood agar, as seen with our bacterial growth. When another set of those two plates were placed in an anaerobic chamber, it was noted how A. faecalis did not do well in an environment without oxygen. Additionally, C. perfringens, E. coli, and the unknown bacterial samples did thrive. However, bacterial growth did result in slow growth that was not as prominent when compared to the results on top.
In this experiment we saw how different types of plates are used to differentiate between different types of bacteria. CNA agar plates are used to isolate gram-positive bacteria since it has antibiotics that go against the gram-negative bacteria. Mango agar plates have high salt concentrations which allow staphylococci growth. Salmonella and Shigella agar plates have bile salts and citrate which inhibits gram-positive bacteria. McConkey agar plates have bile salts and low pH which inhibits gram-positive bacteria. Blood agar tests a bacteria's ability to process blood in a procedure known as homiletic. Bile-esculin agar allows for the growth of enterococci while inhibiting streptococci. EMB agar plate have esolin and methylene blue which inhibit gram-positive bacterial growth. Motility agar tests for the bacterial's motility and their use of flagella to swim away from an inoculation staff. Thioglycolate medium tests for the presence and separation of anaerobes, aerobes, and microaerophiles by a turbidity growth. Lastly, Air tolerance test tests the bacterial ability of using oxygen for growth.